studying sensory systems is a really unique aspect of biology because, every creature sees the world in a different way. We see the world in a different way than a honeybee or a snake, and that really depends upon the biophysical and molecular sensors that we have in our bodies that interpret all these signals from our environment and then convert this information that our brain can kind of reconstruct as our world.
Over the last few decades, we have learned a lot about how we see, smell, taste. Of course, smelling and tasting is all based on chemical sensing, and this is actually the language that most of our cells in our bodies use to communicate with each other. But the somatosensation, which is sense of touch, pain, proprioception, etc., all of these depend on sensing physical stimuli, which is very different. So how do you sense temperature? How do you sense pressure? And translate this into a language that neurons understand, which is an electrical signal, was not known. It was known that these specialized proteins called ion channels exist, but their identity was not known. So my lab has found ion channels responsible for pressure and earlier work in temperature. What your skin is sensing is the indentation. So anytime you touch, your skin is being indented. What's really fascinating is we say there's the sensor in the skin, but actually most of the sensing is happening at this very, very thin, very long projection of a sensory neuron that starts in the back of your spinal cord. This neuron sends processes everywhere in your body, including the skin. And at the tips of these neuroids, that are sub-micrometer in length, sits this tiny, tiny ion channel, which is in nanometers length, that when the plasma membrane, which is the fatty surrounding of a cell that protects the cell, there's this little protein sitting there. And this protein is, as you can see here, is a molecule that comes with 3 identical members in different colors. They come together and sit in the membrane shown in this green color, and they actually bend the plasma membrane. This piezo 2 protein, there's 2 of them, we call them piezo 1 and 2, are proteins that are encoded by DNA, and so a gene. So there's this gene that its only role is to make a protein that sits in a few cell types on the membrane, and its only job is to sense pressure. And when this cell gets pushed or dented, they flatten. And when this flattens, conformation of the protein changes and ions start flowing through this protein. And when that happens, you have the first signal that starts the process that we call touch.
What molecules in our body react to heat and cold?
At least as far as we know, most of these molecules belong to the same family of membrane proteins, these ion channels called trip channels, which are actually first discovered in flies, in fruit flies. But in mammals, a number of them, different ones, and we don't know yet all the ones that contribute to temperature sensation, but at least 2 of the ones that we've looked at, the receptor for chili peppers, for capsaicin, and the receptor for menthol, these TRPV1 and TRPMA channels, they're sort of molecular cousins in the same gene family, and one of them senses heat and one of them senses cold. This is a model of the capsaicin receptor. This hole in the middle opens up like a donut hole, and it allows ions to flow down their what we call electrochemical gradients. So outside there's a lot of sodium ions, and the sodium ions rush into the cell, and the currency of communication in the nervous system is electricity. So when ions are flowing across the membrane like that, it generates an electrical current, and then this excites the neuron. That initial excitation is then carried along the neuron, say from your finger all the way along into your spinal cord. That information gets relayed to another neuron that carries that ultimately to your brain. Your ability to recognize something hot or cold is determined by the biophysical properties of this protein, which is really kind of a magnificent thing, right? Our route to discovering that was a little bit indirect in that we didn't go in first asking what are the molecules that sense temperature. We asked what are the molecules that sense these pungent agents, these natural products, menthol, first capsaicin and then menthol. And with capsaicin we identified this channel, and we knew it was going to likely be important for some aspect of pain sensation, but we didn't really know what the endogenous normal activator, physiologic activator might be. So we threw all kinds of things at it that we thought made sense in the context of pain, like neuropeptides, neurotransmitters, and didn't really find anything that seemed to be consequential. And so then we decided, our pain system does more than detect chemicals. It detects physical forces like touch or temperature. And so we tried changing temperature. Cold didn't do anything to the capsaicin receptor, but heat did. And so we realized that that the capsaicin receptor and the heat receptor were the same. And in retrospect, of course, everybody thinks we made the connection right away because you eat a chili pepper. But it didn't have to be that way. It could just be that they're on the same cells and you activate the same pathway. And so that was kind of a eureka moment when we realized that this is kind of a very parsimonious explanation for your everyday experience. Among sensory systems, I always say, I think the somatosensation and pain is probably the one that's most important for our survival and well-being. Because if you don't have it, you really have trouble learning how to avoid things that can be injurious.